We will have a split presentation today. I will present the theoretical framework, and Attila Szabo, he did the experimental work, so he deserves to present that. Oh. So, split presentation. First half is mine about the theoretical framework, and Attila Szabo, he did the experimental work, so he will continue. And we have another team here of the AYA, Hungarian ayahuasca research team, the heart of the heart, Petra, Bokor. You see that, yeah, microphone. Oh, yeah, I try. Okay, I am microphone shy, as you figured it out. Okay, so for many of you, it may sound the Hungarian ayahuasca research team sounds like South American goulash club, but please don't kid us, okay? Because I will show you, we have a tradition here. Can we? Yeah. So with Louis Luna, we have been making publications on ayahuasca for about 12 years. And you should know that it was a Hungarian researcher who gave DMT to humans in experimental conditions. Of course, not in general conditions, because South American people do that. So Sarah was the first who did experiments on DMT. And for you, you heard about the DMT is a serotonin agonist, but you should know that serotonin agonism is a necessary but not satisfactory condition for psychedelic effect. This is the current view that metabotropic glutamate effect is also necessary, because serotonin itself is not a hallucinogen. So basically, DMT acts on the serotonin 2A and metabotropic glutamate dimer, because the receptors combine, agonizing one and blocking the other one. But there is another more interesting receptor for us, the sigma-1 receptor. And basically, sigma-1 receptor research gave us the opportunity and the idea how to look, after 50 years, how to look for a physiological role of DMT. Because that's the problem, the controversy, you know, that we got so fascinated by the psychedelic effect of DMT that we totally forgotten that probably if God created this ligand, you know, God did not create it for us to get trippy dreams, what many neuroscientists suppose. You know, I know high merit researchers who say, oh, DMT is a hippie drug, doesn't worth any scientific attention. And you know, it was not the case for endogenous opiates. Very quickly, when they discovered that we have endogenous opiates, right on, they started to study the physiological effect. For me, that means that most of the scientists were biased, you know, so because of the psychedelic background of this. So the point is, our proposal, that DMT is a body-expanding drug. So basically, the psychedelic effect of DMT represents an island, and the bodily effect, let's go somatodelic effect of DMT represents a continent awaiting discovery. Here, this side is just, I give a lot of credit to Joseph Ferriglea. I always considered psychedelics an adaptogens. And basically, the topic of this discussion is about the adaptogenic effect of DMT. We had 2 publications within the last year. I present the first one. This set the theoretical framework for the physiological role of DMT. I put the mosaic pieces together, which points toward this direction, that DMT has a somatophysiological effect. So as you see, we split from the mainstream on 2 accounts. We are focusing on the somatic effect, and we are focusing on the physiological effect, not like many others, colleague of mine who consider DMT causing us schizophrenia, psychosis, and so on. The first piece is the enzyme, the INMT enzyme, which synthesizes DMT. DMT is a trace amine. Usually you don't find in the body, but where you find this enzyme, you can be sure that sometimes DMT probably has a role in that part of the body. Where do you have this enzyme in the highest concentration? It's not the pineal gland, you know, it's the lungs. And there are other organs like placenta, which contains this enzyme in the highest concentration. And what is interesting for us, that this enzyme is always expressed, is always there. Just there is an inhibitory ligand which is sitting on this enzyme and not letting it work. I will tell you later the significance of this. And another interesting that the plasma monoamino oxidase does not break down DMT. The gut and liver mode, yes, but the plasma monoamino oxidase is somewhat different, and it leaves DMT untouched. And here is the very fascinating piece of this mosaic. The brain has free active transport for DMT. First, DMT passes the blood-brain barrier by an active transport, not just passively penetrating it. Do you know what compounds pass the blood-brain barrier by an active transport? The very vital glucose, amino acids. And here we are, DMT. So probably it has a very important role, but that's just the first step. Inside the brain, DMT gets into the neurons by the well-known serotonin transporter. Once inside the neuron, it gets into the vesicles through the vesicular transporter 2. So you can see somewhat DMT is very important for the brain. It is concentrated in the synapses and stays there for 1 week. This is what the brain would do for a neurotoxic agent. In many chemical catalog, you see freemetoxy and N-DMT as a neurotoxin. They schedule it that way. So my colleagues are biased, you see. And this is the other important piece. It is about 4 years ago when they discovered that sigma receptor has an endogenous ligand, and DMT is an endogenous ligand for that. And sigma receptor is an intracellular receptor. It sits between the mitochondria and the plasmatic reticulum. So very probably has an important regulating role in the energy mechanism of the cell. And you know very well, Rick Strassman, he raised the possibility in his book that DMT is released during near death, because many features of the DMT effect you can find during the NDE experience, like a life review, the entities, bright lights, so on. So there is a good reason to suppose, not direct, just indirect evidence, that to suppose that DMT is released during near death. So here is our scenario. Putting those mosaics together, I showed you. So here is the story. In response of the signs of agony, the lungs release a huge amount of DMT within seconds. The lungs do not have to synthesize the INMT enzyme from the genes. It's already there, I told you, all life long. It is just waiting, to kick in. The synthesized DMT is dumped into the arterial blood, so the shortest way to the brain really is from the lungs. If I would be God, I am not, sometimes, even if I feel so. So if I would figure out how to flush the brain the quickest way by DMT, I would say, hey, that's the lung, the best source. The plasma mode doesn't break it down. Since the body doesn't have too much time, the heart is kicking the last, so definitely the brain must use free active transport to take up the DMT, concentrate it, and possibly the DMT can exert hypoxia preventing or mitigating effect, so extending the lifespan of the brain, which we suppose, and we have been running experiment for that. You may ask, where is the psychedelic role? Of course, this last line was not presented in a scientific journal. Just this is for you, for internal use. So once the DMT is in the brain and the rescue is not coming, the cavalry is late, so then the TMT takes up the psychopompic role, guiding the soul through the bardos. And there is another scenario at birth. I put down the mosaics here, the pieces from literature, which indicate that DMT must have a role at the time of delivery. Many infants, they can get hypoxia during the delivery process. The INMT gene is part of a gene network, which is important for maintaining the pregnancy. And interestingly, the highest activity concentration of the INMT in the lungs is during the first 2 weeks. But the difference is that because the fetal circulation is totally different than the adult, the source of the DMT cannot be the lungs. Placenta is the next organ, which has this enzyme in high concentration. Basically, you know what we proposing, and we are starting a research series. Our ultimate goal, just imagine this, using DMT in cardiopulmonary resuscitation. Of course, it's far down the road, but this is what we are proposing, you know. Imagine, so someone is in clinical death, IV DMT, or use it for purple infants, or using in coma. Okay, so I give the best first results are from immunology, and I gave it to Attila to present those results, what we find. Okay.
All right, thank you. So I'm Attila Szabo. I was responsible for the. Can you hear me? All right, this will sound weird. So I was responsible for the experimental part of this project, and I'm going to give you a short introduction. I have only 5 minutes, right? It's going to be fast. It's going to be tough because usually it takes 8 to 10 lectures at the medical university to give the basics in immunology for medical students, and I have 30 seconds right now for that. Anyway, as you may already know, that the immune system, the main role of immune system is, on one hand, regeneration and healing, of course. The other hand is protection, right? It can discriminate between harmful and harmless self and non-self signals. And it has essentially 2 branches. Innate immunity, where's the pointer? I need a pointer. Sorry. There is no pointer. Okay, anyway. So there are 2 branches here, what you can see. Innate immunity and adaptive immunity. This one is always there. It's in your genes. So this is the first line of defense. This is which is protecting you all the time. Thank you. All the time. And here is the other one, the so-called adaptive immunity, which is essentially a tabula rasa at the time of birth. Okay, it is currently building up immunological memory. And actually your protection or immunity itself is an interplay of these 2. Actually, these are 2 aspects of immunity, like the 2 sides of the same coin, okay? Now, let's see, what cells do we have here? We focused on a cell type called dendritic cells, these guys here, okay? Because they are so-called antigen-presenting cells. Now, these cells are able to connect and orchestrate these 2 responses, innate and adaptive immune responses. So they are essential in mediating anti-tumor, anti-cancer, anti-parasitic, antiviral, anti-whatsoever, you know, and so on, so anti-pathogenic immune responses. So if you can somehow modulate the activity of these cells, then you can actually alter the immune functions as well. So it's they play a very important role in the mediation of immunity. Now, another important aspect of our studies is the manipulation of inflammation. You know, inflammation is very important because in every 3rd case in the clinical setting, every 3rd case has something to do with inflammation. Either the etiology of the disease or the symptoms are something to do with, you know, directly or indirectly with inflammation. What you see here is, I know it's quite scary, no worries. This is a very schematic figure about the initiation of an inflammatory response. So when there is an invasion of pathogens, or there is a tumor somewhere in your body, or anything that can elicit inflammation, a set of sensors can detect this signal. These are the sensors called pattern recognition receptors. But what's important is that this signaling is converging into the production of the so-called inflammatory cytokines. These are the compounds that are mediating the symptoms of inflammation. So these are the molecules that are causing inflammation in your body. So they are increasing the diameter of the vessels, increase the permeability in the vessels, so you will have the symptoms, you know, this kind of redness, swelling, edema, and so on. All right, now let's put these stuff together. We wanted to check whether immune cells, again, dendritic cells, human dendritic cells, express this sigma-1 receptor, because as just Ede mentioned, sigma-1 receptor is the one which can actually bind DMT. So DMT is an endogenous ligand for sigma-1 receptor, right? So it makes sense, right? So we check whether it's there within the dendritic cells, and we found that yes, they are here. They have very nice level of sigma-1 receptor, and we also checked what is happening when we put these cells into an inflammatory environment. Now for this, we used polyice and LPS. These are artificial compounds. We're just mimicking inflammation with these. Polyice is actually a double-stranded RNA mimic, so you can mimic viral infections with this, and LPS is the constituent of a bacterial cell wall, so you can mimic bacterial infection with this, right? So if you have a look at this figure, you see that nothing is happening. So immune cells, they have a very stable expression of sigma-1 receptor. It is not changing during the course of inflammation. However, what changed was quite striking and very interesting, because when we check the level of inflammatory cytokines, these are the guys which are causing the signs of inflammation again, okay? So I1 beta, IL-6, and so on and so on. This one also, this is a so-called chemoattractant. This guy can recruit more and more inflammatory cells to the site of inflammation. When we applied DMT and 5-MeO-DMT, so we just simply put it in the supernatant of the cells. We used cells isolated from healthy volunteers. We saw that after reactivation with LPS, with this bacterial ligand, or polyice with this viral ligand, we saw that the cells produced significantly less amount of inflammatory cytokines and chemokines. On the other hand, they produce more interleukin-10. What is this interleukin-10? Interleukin-10 is an anti-inflammatory cytokine. It makes the whole thing a bit more interesting, right? So this is anti-inflammatory cytokine, also involved in regeneration, tissue regeneration, because in the late phase of inflammation in vivo in the body, this helps in angiogenesis. So the renewal of blood vessels and the regeneration of the tissue. So what we saw here, it was the actually a complete inhibition of inflammation and a mobilization of interleukin-10 secretion by these cells. To be a bit more realistic, we used a more complex system, and we wanted to have a look at what is happening when we put these dendritic cells together with T cells. Now, T cells are the guys, they are the key players of adaptive immunity. You remember, adaptive immunity is the one which provides you with immunological memory, okay? So your immunological memory relies on the activity of T cells. Now, this is actually the final outcome of the immune responses. And when we used here a bacterial species and a viral species, E. coli and influenza virus, we saw that the number of activated helper T and helper T1 and helper T17 cells decreased significantly. Now, why is it so important for us? Because these Th1 and Th17 cells are key players of inflammation in the body, and we know that these cells are highly involved in pathologic inflammation. Like in the last few years, it's been described that these cells play an important role in the etiology of central nervous system inflammations. Also, they have a very important role in development of Alzheimer's disease, most possibly in case of bipolar disorder, depression, and so on. According to the new paradigm of inflammation in the brain, inflammation contributes to a lot of things. Neuropsychiatric disease, as we have already heard of in Eduardo's presentation. So, neuropsychiatric diseases, neurological diseases, multiple sclerosis, and so on. Now, it seems that DMT and 5-MeO-DMT can very effectively inhibit the activity of these cells, Th1 and Th17 cells, through dendritic cells and through the sigma-1 receptor. And this is the last slide. I really don't want to waste your time with this. This is a very important slide, and we have many more data about this and very interesting data, but just simply don't have the time to explain these. Here I just wanted to show you that when we knock down the sigma-1 receptor gene, a technique called RNA interference, so we silence the gene, so the cells essentially lost sigma-1 receptor, and then we treated the cells with DMT or 5-MeO-DMT. You see here, this effect was absent completely. So it means that the sigma-1 receptor is essential in this, in this process. So the DMT or 5-MeO-DMT is somehow acting through the sigma-1 receptor, but not completely. As you can see here, other receptors must be involved here because there was the original levels could not be established again or re-established after the gene silencing. So there must be at least one receptor here, and I'm quite sure that it must be the serotonin 2A or serotonin 4 receptors, which are also expressed on dendritic cells and immune cells. But that's another question. And this is also about the same stuff with poly-IC activation. And let's take stock in 30 seconds. So what we know here is that, and what we found is that the human dendritic cells express sigma-1 receptor, and if you activate this receptor with DMT and DMT analogs, then you will have a very massive anti-inflammatory effect. And this is very important because our findings, raising the possibility that DMT may have a very important physiological role. So it can be a very important immunoregulator on the one hand, but also can contribute to other physiological processes in the body concerning tissue regeneration or neuroregeneration, and all of these other processes. So these findings are actually pointing out the new physiological role of DMT, excluding the psychedelic role of this molecule. And this is very important because we know that DMT is quite ubiquitous, in the biosphere of the Earth. This is an evolutionary, highly conserved molecular motif. It's present in many plant species, many animal species, almost 100% in the vertebrate species and mammals. So yeah, maybe it's not only there for the psychedelic effects, right? But also it may have other important roles, as we can see. Okay, thank you very much. That was it.